So when the Q&A shows up, will I see any of these questions at all? I know I'm not selecting from them, but I'm just
Yeah,
wondering.
you will, Eric. Dana will try to pull together sort of common themes, so you're not answering the same question 3 times and
All right.
post them, but you'll be able to see them as well.
All right, sounds good.
Good. Okay.
Whoops. All right.
All right. I know we're at 2 o'clock, so let's get started, everyone. Hello and welcome to today's OBSSR Directors webinar entitled Molecular Mechanisms and the Evolution of Vocal Learning. I'm Bill Riley. I direct the Office of Behavioral and Social Science Research here at the NIH. Before I introduce today's speaker, a few housekeeping items to go through. Today's webinar is being recorded, and that recording will be available in about 2 weeks on the OBSSR website. So if you know of folks who are unable to attend virtually today, you can point them to the recording of this that will be available in a couple of weeks. Today's presentation will be followed by a Q&A session. All our phone lines are muted during the webinar, so there's a question and answer area that we'll take questions from. So you can use either the Q&A or the chat feature to do that. And so just click Q&A and select all panelists and put in your question and send it. Dana Green in our office will facilitate the question and answer session and ask your questions to the presenter at the end. So I'm pleased today to introduce today's presenter, Erich Jarvis. Dr. Jarvis is professor and head of the Laboratory of Neurogenetics of Language at the Rockefeller University and an investigator at Howard Hughes Medical Institute. He's also an alumnus of Rockefeller University. He returned to campus there in 2016 as a tenured professor, heading up their Laboratory of Neurogenetics of Language. Dr. Jarvis investigates vocal learning in songbirds and other animals as a model for understanding spoken language in humans, and his research integrates computational, behavioral, physiological, and molecular techniques to explore the neurogenetics of vocal learning and the evolution of this complex behavior. He's, as I said, an investigator at Howard Hughes Medical Institute, been there since 2008, and a recipient of numerous awards, including the National Science Foundation's Alan T. Waterman Award, and NIH Director's Pioneer Award, and a number of other awards as well. So it's my pleasure to welcome Dr. Jarvis to present today on molecular mechanisms and the evolution of vocal learning. Erich, thank you.
All right, thank you, Bill, for that introduction, and thank you to you and others for inviting me to give this presentation. I'm excited to talk about some of the latest things we've been working on and to hear feedback throughout the question and answer sessions or emails you might want to send me later. So yeah, the title of my talk here is Molecular Mechanisms and Evolution of Vocal Learning, because I think when you study the two hand in hand, you can understand how nature does things naturally as an experiment in a way to help you understand mechanisms, how complex behaviors work. In this case, vocal learning. It's a very rare trait. Only few groups of animals have it. Some of them shown here and their close relatives that don't. And so I'm going to talk about this from 3 perspectives: the behavioral, the anatomical, and the genetic levels of biological analysis, and then end off with a new hypothesis that we're testing from this work using mice. And so starting at the behavioral level, and as I go through this, I know some of this will be fast, some of this will be in detail that's hard to understand. And if you want to find out more, I encourage you to read a review article and perspective article I wrote in 2019 in Science, and there are other articles as part of a special issue on language I encourage you to look at as well. So when we humans think of language broadly and spoken language in particular, we tend to experience this as a unitary trait. But in practice, in actual real biology, it consists of multiple components. And some of those components that I've colored in blue here in this circle, like auditory learning, are common among species of vertebrates, and some are actually pretty rare, like this vocal production learning. And all of these traits together, the syntax, the pragmatics, and so forth, come together to give us the experience of what we call spoken language. So with this definition, in mind, I like to define spoken language as a specialized form of learned vocal communication with some components found in most vertebrates and a highly specialized vocal learning component found in only a few species. And who are they? There are 5 groups of mammals and 3 groups of birds. So that's us humans, of course, but dolphins and whales, these are cetaceans, bats, elephants, and seals, as well as songbirds, hummingbirds, and parrots. So all 8 of these groups of species can imitate sounds and pass on those imitated sounds culturally from one generation to the next, like we do for language. Um, and they all have close relatives that cannot. So it's thought that all of these species evolved this ability independent from a common ancestor, that is de novo. In contrast, we depend upon auditory learning to hear the sounds that we're going to imitate. But the ability of auditory learning is ubiquitous amongst the animal kingdom, at least amongst vertebrates. And the common example I like to give is your pet animals. Like you can teach your dogs to understand the word sit in English, siéntese in Spanish, osawari in Japanese, and so forth, in multiple languages, or whole sentences like roll over, get the ball, get the toy, get the newspaper, and so forth. But a dog can't say, okay, you got it, I will sit. Instead, a dog usually goes woof, right? And that woof, it can have some modification, but it's mostly innate. And it can learn that woof in different contexts, but the actual acoustic structure and the sequencing of the sounds are innate. So uh, now, not all vocal learners are equal. There are some that are more complex than others. Of course, humans are the most advanced, but after humans comes the parrots. And here's an example of a small parrot called a parakeet, a budgerigar, Disco, who was raised with humans for 4 years.
Never shake a baby bird. That would surely be absurd. Shake a baby bird. Surely be absurd. It's cheeseburger. Stormtrooper. This guy used to be a spy.
So what's going on here is that Disco has picked up about 400 human words in his lifetime in the 4 years that he was living at that time. And what's remarkable here is not only imitating human speech words, but recombining them into new sentences. Uh, Most of the times, those don't make sense to at least to the owners. Maybe they make sense to Disco. But a lot of times, like seeing the dog here and Disco goes meow, kind of getting the category correct, but the actual species not quite correct. A lot of times Disco is getting it right. So there is decent vocal learning going on here. Now, some of our closest relatives, people get excited about what non-human primates can do, but they can't do what Disco does. And here is the most that you can find.
How about when you're coughing? That was good. You did a sneeze and then a cough. Excellent.
Yes, now that is excellent for Coco. Coco is a gorilla, female gorilla, raised with humans for 39 years at this time, and had voluntary control of, let's say in this case, of air going through the vocal tract, like coughing here, but mostly able to produce voiceless sounds, that is not modulating the sounds with the larynx. And so Coco has good auditory learning though. Coco could understand several thousand human words, but can't produce one of them. All right, so this has pondered myself and many other scientists to say, why can't we get some of our closest relatives to say something as simple as apple, but yet, they're like 300 million, I mean, 6 million years distant to us, but yet our distant relatives like these parrots, 300 million years from us, from a common ancestor, can say things like, not just apple, all kinds of varieties like golden delicious and so forth. And that's the question I've been pondering actually for my career. And I think we have some explanations why. And I'm going to tell you some of them at the level of the anatomy that led to what we call a motor theory of vocal learning origin. So, but what we're proposing is not the only theory out there. And here in this review, I looked at 6 of them in the literature. And I just want to quickly go over what they are so that at least you have a sense of what people have proposed. Number one here is that there's a general difference in the brain, the size of the brain. So green here is basically emphasizing the forebrain. Red is the brainstem motor neurons for vocalizations. And blue here is the actual vocal oral facial musculature, and that produces the sounds. And so one theory is that there's a bigger brain in the vocal learners, and that bigger brain allows the development of vocal learning language circuits, or there's more neurons. The other is that it's not necessarily in the brain, it's actually in the muscles or the vocal organs themselves, in particular being further descended in humans or having more musculatures, different types of muscles in the vocal learning birds. The other is that it's not the muscles or the big brain, but it's actually the presence or absence of a circuit that controls the vocal motor neurons in the brainstem. Another is that those circuits are there in all these species, but it's what differs is whether that circuit makes a direct connection to the motor neurons for vocalizations or an indirect connection or more enhanced one in red arrows here. Another is it's not the direct or indirect connections to the brainstem, but it's the auditory input from auditory cortex, secondary auditory cortex into Broca's area or laryngomotor cortex, where it's argued that in humans there is a direct projection or an enhanced one here in red that you don't find in non-human primates. Or finally here, it's many of these species actually have auditory and vocal motor circuits that are similar to each other. What differs, particularly in humans, proposed by the linguistics experts, is that there's a separate language module in the brain that makes all these algorithmic computations for language. And it's that language module that feeds into auditory and motor circuits to control spoken language, and some might argue vocal learning. And long story short, when you really look at the hard evidence in the literature, I actually think it supports a combination of 3 of the 6 here. And that is, in humans, yes, a bigger brain supporting more space for speech and language circuits. Our songbirds and parrots actually having more neurons per millimeter squared. And presence or absence of certain circuits that do make direct or indirect projections to the brainstem motor neurons. And what I'm going to do now is show you some of the evidence for some of these that then led up to this theory of origin of these circuits. One is beginning back actually when I was a postdoc, discovered that when songbirds sing their learned songs, like this canary here, that act of singing is associated with a robust increase of what we call activity-dependent genes or immediate early genes seen in white here. This is the messenger RNA product of the gene. And the red stain here is the crystal violet staining of all the neurons in the brain. This is the front and the back of the brain. And we can see from this bird who sang in a half-hour period, you see a robust increase of gene expression in the very brain regions that are controlling the learned singing behavior. Like in this area exon nucleus right here, there's a 1-fold increase for every song bout a particular bird sang in the last half hour before looking at the brain gene expression. And this was a motor-driven gene expression response. That is, it didn't require the bird hear itself sing. It didn't require somatosensory feedback from the muscles. It didn't require that the bird was seeing another bird communicating with it. It required that the act of singing of learned sounds, not innate sounds, be performed. And we use this, what we call a behavioral molecular mapping tool, like an MRI tool, to map vocal communication circuits across multiple bird species, shown here. And we found that all species that we looked at, whether they be vocal learners or not, had the auditory pathway in their forebrain, colored in blue here, which is consistent with all of them having auditory learning. But only the vocal learning species, the ones who can imitate sounds, had regions in their forebrain, colored in red and yellow here, that you don't find in the non-vocal learning species. And in songbirds, we know these yellow labeled regions are responsible for producing the learned sounds, and the red labeled ones form what we call a cortical basal gangliothalamic loop that's responsible for imitation of the sounds. And so, so this indicated what was remarkable. There were 7 such regions in the brains, the yellow and the red here, of all 3 vocal learning bird groups. So it indicates that either they evolved this trait independently, remarkable convergence, shown by these red dots here, or maybe there was a common ancestor back here sometime 60 million years ago, and there were 4 independent losses. some type of constraint to maintain this trait. Or maybe everybody has it to various degrees, and it's been independently amplified in an evolutionary perspective in these vocal learners. Or maybe the tree is wrong, the phylogenetic tree. And at this time, I was a hardcore neuroscientist, but not a phylogeneticist. And many people, when they see this, they're arguing, no, these species got to be closely related to have that much similarity. There was an alternative view as well. Some actually took these pictures and put them in religious texts to indicate that it supports intelligent design. So I got involved in a big phylogenomics project, which I ended up helping to lead, called the Avian Phylogenomics Project, where with next generation sequencing technology at the time in 2014 or so, we sequenced the genomes of species representing all bird orders, including vocal learners with the red dots and their close relatives according to different phylogenetic trees in the open circles. We ended up publishing a number of papers from this effort, one of them being this redoing the phylogeny of birds. But instead of using just a few genes or genome hybridization results, we looked at the entire genome, and we got, for the first time, a genome scale phylogenetic tree, in this case of birds, and we asked, what are the relationships on this new tree? And we find lots of rearrangements in those relationships. What about the vocal learners? Well, actually, they still are separate. In fact, the hummingbirds are pulled much further away from the songbirds and parrots, according to the older tree that we were all using. And so it does support at least 3, if not 2 independent gains, followed by maybe several losses. If you had to have a common ancestor, it would have been at the time here of the mass extinction of dinosaurs, with just birds remaining, few surviving who had vocal learning, and then 10 losses in the last 65 million years, or at least in a 30 million year window of time, which we don't think is the case. So, so we think it's independent. What about with humans? Well, we start to compare songbirds to humans, and we find the first thing we see is that here's a zebrafinch brain to scale with a human brain. Size doesn't matter. You can fit 3,000 zebrafinch brains into a human. Neither does cortical folding or surface cortical regions matter. What I argue matters is the presence or absence of a specific circuit. But to get at what that circuit looks like, we had to reevaluate a dogmatic view that had been around for over a century on vertebrate brain organization, beginning in the late 1800s, early 1900s, where the founders of comparative neuroanatomy argued that the brain evolved in a linear fashion. They took Darwin's view of evolution and Aristotle's view of scala natura and their own views, and even religious and racial views, and argued that in the vertebrate brain, starting out with fish, you have these basal structures called basal ganglia that they named, paleostratum, archistratum, and so forth, that were passed on from one lineage to another, from fish to mammals to reptiles and birds. And birds had, all in purple here, just one large basal ganglia with minimal cortical tissue, the hippocampus here called archicortex at the time, and the olfactory bulb called paleocortex. And I organized a consortium of neuroscientists who evaluated all the evidence and pulled together their own evidence. By the way, they were even arguing the neocortex at the time, but that's what they named it, neo, for being new in mammals, was biggest in Europeans, followed by smaller in Asians, and then smallest in Africans, and then all species below that. And so he argued that this view doesn't make sense, looked at all the evidence, and came up with this new Martin view, where the bird brain and the vertebrate brain in general has a large cortical territory in green here that in mammals is layered in its cellular organization, but in birds and other species, it's clustered in its organization. And here is the true basal ganglia in purple and blue here that matches and is homologous to the basal ganglia here of humans. And so putting this new view together, now we can compare to humans and songbirds more accurately, the vocal learning birds. And the model we came up with and updated more recently in this 2019 paper I told you about is that I argued that all species of vertebrates have a complex auditory circuit like Wernicke's areas of humans. And this is why dogs can understand the words sit, siente, say, come here, roll over and get the ball, and so forth. And why Coco understands 7,000 human words. I mean, 2,000, sorry. And I argue that it's inherited from a common ancestor with other vertebrates, including these birds, showing here in the blue colored label regions. However, relative to non-human primates and other mammals, we can only find in humans, labeled in yellow in here and red, these regions that are responsible for spoken language acquisition, speech, and that they are functionally similar to the yellow labeled regions, the motor pathways for producing speech or song in these birds, and the vocal learning pathways for imitation in red, with auditory input going into them, showing by this blue arrows. And so how could this come about? How could you get such convergence, such similarity? There are differences across species, but a lot of the similarities outweigh some of these differences. And we didn't really have an answer until we found that not only the vocal learning brain regions have activity-dependent induction of these immediate early genes, but so do the rest of the brain, the surrounding brain circuits, in this case for movement behaviors, where a bird is hopping in a rotating wheel for the first time in its life. And we find that all 7 of the brain regions and all 3 of the vocal learning birds have motor-driven gene expression regions around them. And the non-vocal learning species also have them, but without the holes of song nuclei inside of them. And when we look at the connectivity of the vocal learning circuits and the surrounding brain regions, the connectivity is similar. We have this cortical basic thalamic loop, and we have descending motor circuits, in this case to the wing or the head, the leg, and other motor neuron circuits. There are some differences. The surrounding motor neurons don't necessarily make those direct projections that allow you for fine motor control of movement. And they also make collaterals to the striatum, like layer 5 neurons of motor cortex in mammals, but you don't see that in the vocal learning circuits, at least in songbirds. So putting all this together, well, we also found, and I didn't say we, another lab, Stephen Brown's lab, found that when he gets dancers to do complex choreographed sequence movements and images their brains, and finds that the brain regions that show the highest activation with learned sequential movements are directly adjacent to Broca's and laryngomotor cortex regions. That is, they're like in these songbirds, the learned motor circuit is directly adjacent to the vocal learning circuit that controls song or speech. And so putting all this together came up with called the motor theory of vocal learning origin, where I argue that all vertebrates have a brainstem circuit involved in production of innate vocalizations, like the dog's whiff or the chicken's crowing. They all have motor learning pathways for producing learned movement with the black arrows here, and for learning how to move in the white arrows here. And I argue that during embryonic development, this motor learning circuit is replicated multiple times in the vertebrate brain, and in humans and in song learning birds, through some mutation, it created an extra brain pathway duplication that got hooked up to the brain circuits that control vocalizations and took it over to get this emergent vocal learning circuit from the surrounding motor pathways. And if this were true, we would expect the genes that function inside the vocal learning circuit would be similar to the surrounding motor learning pathways, with some differences that control differences in connectivity or rapid integration of auditory input with motor output. And so this is what we set out to test in my topic 3 here, on the genetic and molecular mechanisms of song and song learning birds and spoken language in humans. And one of the initial experiments we did to get at this is we laser dissected the song learning nuclei in each of the vocal learning bird groups and the surrounding motor regions. And we did the same thing for non-vocal learning species, where we would expect to find vocal learning circuits if they were there, and profiled them to microarrays at that time. So these are glass slides with oligonucleotide spots for thousands of genes that we analyzed from the genome. And the Allen Institute for Brain Science was doing something similar for human samples, postmortem brains that people donated to science, from 6 people and also from 6 monkeys, from all over the cortex, the basal ganglia, the cerebellum, and so forth, and profiled them to microarrays as well. And students and postdocs in my lab at the time took this data, generated some of it as well, and did a lot of computational analysis to ask the question, can we find in songbird brains gene expression specializations, up or down regulation of one or more or many genes in these vocal learning circuits compared to human speech areas and the surrounding areas as well. And looking at 7,000 orthologous genes from these genome alignments, we found that The area X song nucleus of songbirds involved in song learning match in specialized gene expression a part of the anterior human striatum, not other areas of the striatum, not the cortex, whatever, but this particular location in the anterior striatum for the same set of 75 genes that are specialized out of this 75, out of 7,000. And we found that the RA song nucleus matched a set of 55 genes with specialized expression of up or down regulation in the human laryngeal motor cortex. We didn't find any specializations that match for HPC and other song nuclei in songbirds, nor did we find, even for RAX, in the striatum of non-vocal learning birds or of non-human primates or in the cortex, regions that showed specializations with humans. That is, this was specifically shared between the vocal learning birds and humans. And when we looked at these genes, shown here in a heat map, each row is a gene. Each column is multiple samples from different species, like a songbird, a parrot, and a hummingbird, and the non-vocal learning birds. We can see the pattern of blue or red for down or up regulation of these genes is similar across all three vocal learning bird groups and humans here. And many of these genes in the laryngeal motor cortex region here are downregulated. That's why you see the blue, and they're involved in axon guidance functions. The ones that are upregulated in red here, like parvalbumin, are involved in neuroprotection, calcium buffering, and so on. And we didn't see this in other cortical regions or non-human primates. Looking at one of these genes, SLIT1, an axon guidance molecule, here showing by in situ hybridization its downregulation in the RA song production nucleus of all vocal learning birds, but not of non-vocal learners. And here showing the white signal here for the human brain surface map means downregulation. And in two regions, dorsal and now what we call ventral laryngeal motor cortex. Two regions, we also found two regions in parrots, by the way, which we think is two parallel vocal learning circuits. And these two regions here match precisely in coordinates with Eddie Chang's group has found shows high neural firing rates when he is examining patients and trying to avoid speech areas for epileptic or tumor regions. The highest spots of neural firing during speech production in humans in the primary motor cortex. And so we got this far with these genomes and we're all excited. We got a set of 50 to 70 genes that we wanted to study. And students and postdocs in my lab, we begin to try to study these genes, trying to manipulate them in songbirds and in mice. And we soon found out that the genome assemblies were highly fragmented. Here is the gene with lots of gaps here between SLIT1 is like 35 exons here. And we had lots of gaps, missing parts of genes, incorrectly assembled genes. And what we were finding is that some of these vocal learning related genes had repetitive regions in their promoter regions, only the vocal learners, which made it difficult to assemble them. And this was causing many months to clone genes, to correct them, and so forth. And many of us were working with artifactual gene structures and making false conclusions about the biology that we weren't even aware of with these draft quality assemblies. So that prompted myself and also leadership of what was called the Genome 10K Consortium at the time to say, Erich, can you lead this Genome 10K group, which I later we called the Vertebrate Genomes Project, and do what you did with the bird genomes now with vertebrate genomes broadly. And so I took on leadership. We changed the name to the Vertebrate Genomes Project with the mission of not just 10,000 species, but eventually can we produce genomes of all 70,000 vertebrate species on the planet? At least that's the long-term goal. And not only just to generate the genomes, but seeing what's happening in comparative genomics, we needed to compare 40 species. If I'm going to correct genes for 40 species, one at a time, that's going to be too much. So we set our goals to generate one high quality, error-free, gapless, chromosomal level, and phased haplotypes, maternal-paternal chromosomes, annotated genomes for all those species to address the fundamental questions we'd like to address. And we took 16 species, including multiple vocal learners, came up with a pipeline that requires long sequence reads, like from PacBio or Nanopore, optical mapping and other scaffolding tools to take these, what we call contigs, continuous pieces of DNA, assembled DNA with no gaps, hopefully no errors, put them together into longer chromosomes, manually curate them to correct any errors. And here is just show you the size distribution of the different kinds of sequencing technologies we use from shorter molecules to chromosomal length molecules of bats and songbirds and parrots and hummingbirds, my favorite vocal learners, and others that people were interested in had money to support. And we published a special issue on this recently. I don't have time to get into all the details since this is mostly a neuroscience presentation I'm giving to you, but I encourage you to go look at that. And please, when you're considering doing genomes, consider working with us because I think you're going to like these higher quality genomes better. So, but just to show you one result here, here is the contiguity, the length of the molecules of the assembled genomes, the millions of base pairs, when using long reads versus short reads. They're hundreds of times magnitude bigger. Here we find this is all the inversion or join errors that were made in the assemblies using short reads versus long reads here. So we get much fewer errors. Here is a misjoin. We can look at what's called the high-seq plot. Each box here represents a chromosome, and that's amazing. We can even talk about chromosomes with these assemblies without having a linkage map. We correct some of these manually, and we can get chromosomes here, size scaffolds that match the karyotype chromosomes here almost in a one-to-one fashion. For this case, the Anna's hummingbird, a vocal learning species. And so putting all this together with these new genomes now, with these high quality genomes, we ask this question again. Can we find more convergence between these songbirds and humans? And the answer is yes, but even before we got there, we had to reorganize our understanding of the bird brain even further. Over those years, we were doing a lot of comparative gene expression profiling, and more recently with RNA-seq data, our new understanding of the bird brain, we even changed further. All these regions here are argued to be either cortex or cluster amygdala homologs, on the blue versus the red and the green and the yellow regions. We found that all these cell types are actually similar above and below the lateral ventricle space here. And there are really four major cell populations in the bird cortex, so to speak, that we think might be consistent with the 6 different layers of the cortex. And so, and you'll see that show up in a minute. So with this new, even more modernized view of avian brain organization and RNA-seq data from birds and still some microarray data from humans, we found, we validated our prior discovery that area X is convergent with a part of the anterior striatum involved in human speech productions, which was since shown since we published that earlier study. Not just 70 genes, but 180 to 100 genes, depending if it's the anterior putamen or caudate. RA does match particularly the dorsal laryngeal motor cortex. And we also found a match for HVC this time around with more genes and genomes and more brain regions profiled, also to the dorsal laryngeal motor cortex for about 69 genes. And when we put ventral and dorsal laryngeal motor cortex together, we get 250 genes for HVC and 154 for RA. And we found also the L-Mansol nucleus matched some of dorsal laryngeal motor cortex, but it was all the shared genes amongst the cell nuclei. So we think it's not as specialized as the other brain regions. And so what's going on here? 2 brain regions in birds matching 1 brain region in humans. When we look at those 2 brain regions, HVC and RA, and look at the heat map of these genes, we find that, yes, there's some genes in both HVC and RA that are upregulated or in blue downregulated here. But most of the genes in HVC, most of the genes in songbird RA are specific to HVC or specific to RA in its match to human laryngeal motor cortex, indicating possibly different cell types. And the Allen Brain Institute also had profiled different cell types in the human primary motor cortex. And we looked at these and other genes and found that the HVC genes that were specialized in songbird HVC and human laryngeal motor cortex were of the layers 2 and 3 and layer 6 type of neurons in humans. Whereas the RA neurons that had specialized gene regulation specifically in speech areas were in the layer 5 neurons. And so these did match different cell types. Recently, a work came out from Michael Brainard and Todd Roberts' lab, some colleagues of mine, looking at different cell types in HVC and RA cell nuclei, and we compared our bulk RNA-seq data up here in the upper right to their data and confirmed this, and further found that in HVC, The neurons that have the most specialized convergent change with humans are the neurons that project from HVC to RA, the intratelencephalic neurons that go from one cortical region to another. Whereas in RA, the neurons that have the most specialized gene regulation with human laryngeal motor cortex were the neurons that project out and make that direct projection to the lower motor neurons that control the vocal organs. So this was quite remarkable. Now we have genes that are specific to different cell types. What is causing these genes to become specialized in the first place? And a graduate student in my lab, Lindsay Caton, who just got a PhD, dissected out the song learning nuclei in the surrounding regions of these different bird species and profiled their ATAC-seq peaks. These are open chromatin regions of the genome that are poised to regulate genes. And she found, like, for the immediate early genes that are regulated inside or outside the vocal learning circuits, had peaks of ATAC, open chromatin signals in the front of the promoter regions of these genes, like EGR1, whether you're in a vocal learning circuit or not, or even in the liver, which is consistent with their known regulation. However, genes like an androgen receptor that was specialized in HVC had ATAC-seq peaks shown in gray here, only in the genomes that were inside the song learning nucleus, but not the genomes of the neurons outside, in front of the androgen receptor. And this prompted us to look at the human data, and remarkably, we found also the androgen receptor is higher in human speech motor cortex regions than the surrounding motor circuits, which is quite remarkable. And that's the case in both men and women. So we don't think that's a sex-linked trait. And so that's just a few example genes. Now looking at all the genes that are specialized in the song learning nuclei and their regulatory peaks in the entire genome, what we find is whether it's in HVC, RA, or area X song learning nuclei, they are all enriched. That is, genes that are specialized are enriched for ATAC-seq peaks that are also specialized. Whereas the genes that don't differ between the vocal learning circuit or the speech circuits in the surrounding brain regions don't have ATAC-seq peaks that are enriched in those genes inside the song learning brain regions. And we look for transcription factor binding sites in these peaks here that are different between the song nucleus and the surround. And we found roughly a set of 8 transcription factor binding sites, 4 of them shown here, the top 4 rows here, that are enriched inside these regulatory peaks. And one of those transcription factor binding sites for genes that are upregulated in the song learning nuclei, those genes, 50% of them have an androgen receptor binding site in front of their promoter regions. For HVC, 10% for RA and area X. And what about genes that are turned off in the song learning nuclei? Well, 50% of them have the NeuroD6 transcription factor regulate open chromatin region in front of their genes that are there. And what about the immediate early genes that are regulated inside and outside the song nuclei in equal fashion? Well, they have ATAC-seq peaks that have the immediate early gene response, the EGR1 transcription factor binding site in front of those genes. So it's consistent with the in situ hybridization. What about gene function? Whether you're looking at the biological functions or where the genes are located, what's interesting, shown here for area X, is you have a combination of genes that are up inside the song learning nucleus are enriched for a number of things like neural differentiation, axon guidance, and so forth. But so are genes that are turned off in the song nuclei and enriched in the surrounding brain regions shared by all vertebrates, also have enrichments for some of these same functions, indicating that what has changed inside these song learning nuclei and speech regions is the category of gene that has changed, just switched another gene to be a different axon guidance molecule, turn it on or turn it off. What about function, the phenotypic function of these genes? When we compared these specialized gene patterns with the human phenotype database, we found the top hits were involved in speech production. That is poor speech, language impairment, neurological speech impairment, autistic verbal deficits, and so forth. And not running, walking, or something else, but language, speech deficits. For it's only the genes that are upregulated in the song learning nuclei here. So that's very interesting. So looking at them, why upregulate or downregulate some of these genes? And here's just more an example. How does it happen? Taking one gene, NeuroD6, and looking at its ataxic peaks in front of it here, and asking the question, are these sequences where this peak exists here in the highlights here, in front of the gene, does it have change in the nucleotide sequences in the genome? And the answer is yes. In NeuroD6 and all the vocal learning birds, songbirds, parrots, and hummingbirds, there is a region that has accelerated evolution compared to all the other species that are not vocal learners. And that is blown up here. What we see is that the songbirds, parrots, and hummingbirds have a mutation that converts this very homologous sequence across birds from one of what we don't know what it does to an androgen receptor binding site. Subausines, the close relative of songbirds, it's like the chimps of the songbird world, have an extra mutation that prevents it from being a testosterone androgen receptor binding site. So we think there's something going on importantly here about the androgen receptor, and here is our current working hypothesis. We think the vocal learners have independently obtained mutations here in blue lines, which allow certain transcription factors, or like the androgen receptor here, to bind to this regulatory region, make it a new regulatory region, in this case, to turn off NeuroD6, transcription factor expression in the speech circuits and the song circuits. And when you turn it off, that transcription factor can't regulate other genes. And some of those other genes that are already known as targets of NeuroD6 include the ones that are specialized in the song learning circuits, like SLIT1. So we think this is what's causing SLIT1 to be shut off in the vocal learning circuits. But if you mutate the transcription factor binding site in the entire genome, how do you specifically regulate it in the song and speech circuits and not the surrounding brain regions? Well, you code it with histones. And this is what we think is making the open chromatin difference in the peaks there. You coat the surrounding motor cortex regions with histones, but the speech circuits you leave open, so you can modulate the genes specifically in the speech circuits. And so that's the hypothesis we're working on. And there are, looking at all the categories of genes that have these functions, there are roughly 26 genes with convergent expression between humans and the vocal learning birds that have these ataxic peaks, have associated accelerated evolution, and associated with also speech motor function. And many of them are transcription factors, as opposed to downstream molecules. And in the future, just so people know, we're going to focus on some of them, like androgen receptor, ZEB2, which I'll talk about in a minute, NFX1, that transcription factor, plexin A1, it's actually an axon guidance receptor, SLIT1, I told you about before, it's ligand, and one of them is a glutamate receptor, GRIN2B. And so ZEB2, here's an example of one of those genes. It is upregulated in the laryngeal motor cortex, and we think it's in layer 3 neurons. And children with mutations of this gene have a particular speech deficit, other deficits as well, but speech is affected a lot. And here, I'll just give you an example. This is Bella, 11-year-old child. She's trying to say bubble, and she has difficulty producing speech even beyond one word, but she has coordination enough to put the bubble thing in the blower. And so another one is, and we'll be studying that, another one is GRIN2B. It leads to more deficits. And here we started testing when we didn't have high quality genomes. We actually took this human homolog here, and we find that GRIN2B is downregulated in the song learning nuclei after the critical period of song learning occurs in the cortical song nuclei. And it's more quickly downregulated in the song learning nuclei compared to the surrounding motor regions. That's work from another lab. And what we did is took the human NR2B gene, hooked up to a green fluorescent protein, overexpressed it in L-man of adult birds to see if it can reopen the critical period. And what we found is that the more overexpression we were able to generate in the L-man song nucleus, the more variable the song became in these adult birds when it's normally not variable. And red is the normal low levels we would see. And we all unfortunately had a side effect here. These birds also began to stutter. here shown on these syllables here, particularly when they're communicating with other birds, like with directed singing. And so we think maybe we have to manipulate multiple genes or overcome the stuttering, but nevertheless, we are able to get this increased variability. And what about these genes that control these connections to the brainstem motor neurons? What we would like to do is say, for example, downregulate SLIT1 and see if we can make a direct projection indirect and block vocal learning in these song learning birds. Conversely, upregulate some of these axon guidance genes, see if we can make a indirect projection direct, take over an existing motor learning circuit, and maybe induce some form of vocal plasticity or vocal learning. And we'd like to do that in songbirds, but the genetic tools there aren't as advanced, but they are more advanced in mice. And we started thinking about this question, can we test some of these hypotheses in mice? And that's where I'll end off with here in the last part of my talk, testing some hypotheses with these genes. But in order to do that, we needed to first understand the communication circuits of mice. And what was surprising, Tim Hawley's group found back in 2005 that mice have, particularly males, have these ultrasonic, what we call now songs, that they use to courtship females. And here's what it sounds like and looks like. That's a sonogram at the bottom. It sounds like a songbird, which really surprised many of us. And this prompted my group and many other groups to look at mice vocal communication because we always assumed it wasn't learned. And guess what? We found out, even though it sounds like a songbird, it's mostly innate. Some argue totally innate. My group, we argue that it does have some low level of plasticity, but it's not the same as songbirds. What did surprise us equally more more so, is that we found when they actually produced these ultrasonic songs, there were areas of the motor cortex that lit up with these immediate early genes like we saw in songbirds, which we did not expect to find. It was argued that mice have no cortical region controlling vocalizations. And when we looked with transsynaptic tracers, starting with the muscles here, we found that this same area of the cortex was the only region of the cortex besides something here in the insula that had backfilled neurons from this transsynaptic tracing, second order neurons, blown up here in layer 5 neurons, the types of neurons that makes projections to motor neurons. And when we place tracers into this part of the motor cortex here, we found that those layer 5 neurons made projections down to the motor neurons here in brown that control vocalizations. Except in mice, it was 2 or 3 axons per motor neurons, whereas the data in songbirds and the limited data in humans indicates it's hundreds of thousands of axons going down to the motor neuron. So it looks like a difference of degree as opposed to absolutely there or not. And this idea conforms with Roger Lemon's idea that you see increased manual dexterity of different species, leading up to humans. The more axons you have coming from the primary motor cortex, synapsing directly or indirectly onto the motor neurons that control hand movements. And maybe the same thing here exists for the vocal pathway. And it's been long proposed that this is a fundamental difference between humans and non-vocal learning species, is that you have this direct projection here from the primary motor cortex to nucleus ambiguous in humans, also the indirect one, but non-human primates only have the indirect one. But in mice, we found the sparse one, and I'm predicting, and Peter Strick's lab, I think, is going to show this, that there probably is a sparse projection in primates as well. And so Christina Simoni says, I don't believe you unless you stimulate this brain area and shows that it causes laryngeal muscle contractions. And a graduate student in my lab, Cesar Vargas, just recently did this in the mouse primary motor cortex region here highlighted in red. And we found 2 regions that when stimulated with electrodes in the brain causes laryngeal muscle contractions. One here in the putative laryngeal motor cortex I just talked about that has those layer 5 neurons with a 10 millisecond delay, indicating mostly in indirect projections, but a surprising one that we didn't expect and still a mystery to us. In the oral facial motor cortex of mice, you stimulate there, you get a 5 millisecond delay for the laryngeal motor cortex, as well as a very robust response in the jaw muscles here. Indicating maybe a primordial dorsal and ventral laryngeal motor cortex already there in mice. When we lesion those brain areas, though, mice can still sing their songs, but you lose some modulation. What about the gene expression specializations? We find here's an example again, SLIT1 downregulation in the songbirds. we don't find such downregulation or upregulation of these different genes in the mice brain. And so this led us to what we call the continuum hypothesis, where we can start out with lizards who don't vocalize at all, can have vocal learning, whereas mouse and chicken and so forth have limited vocal plasticity. Bats and songbirds, your more advanced complex vocal learners, and finally to parrots and humans, the more advanced vocal learners. And the more advanced you get in this continuous step-like function, the fewer and fewer species you have, as opposed to being a totally dichotomous trait amongst vertebrates. And so going forward, this leads us to think if perhaps if mice have this limited vocal communication circuit in the forebrain, antecedent of what you find in humans and songbirds, maybe we can enhance it with the gene expression profiles we see in humans. And the idea to actually do this, when I first proposed it, a few years later was reported by Yoshida's lab, where they actually took plexin receptor, which is also downregulated in layer 5 of humans, but not mouse. They argued that this receptor may control the direct projection to motor neurons in the human brain. And by downregulating it, you can actually induce it. And that's what they did here. Here is a wild type mice, and here is a mouse with downregulation of this receptor in the layer 5 motor cortex neurons. And you can see the red little spots here indicate more axonal projections to the, in this case, the limb motor neurons that control movement of the hands. And these mice showed greater manual dexterity of their hand movements when eating spaghetti. And so can we do the same thing for vocalizations? And what's the model that we're working on is why turn off some of these genes? Why lose function? Well, genes like plexin or SLIT1, when they bind to the receptor, in the case of plexin, it's, SLIT1 is robo, when they bind to their receptor, like in the brainstem motor neurons, it will repel those axons away, most of them. However, and you'll get just a sparse projection. However, if you downregulate the SLIT1, we think even with the receptor in nucleus ambiguous, it allows for a direct projection to form and for the motor learning circuits to have more direct control of the motor neurons that control muscle movement, in this case for vocalizations. And recently, Lomax Boyd, a postdoc in my lab, was successfully downregulated SLIT1 in the motor cortex of mice, and these mice showed increased rapid ability to modulate the pitch of their vocalizations compared to wild type. The heterozygous is intermediate between, with the wild type more fancier. So, and we can see that in the knockout mice, they have greater axonal innervation in nucleus ambiguous compared to the wild type mice here. So this is the first time we're actually able to get mice to really modify their vocalizations with a condition that we find in human genomes. Whoops, that's for me to stop. Okay, I have 2 more slides. So, all right. Here, SLIT1 also binds, is regulated by another gene called FOXP2 that when mutated causes a speech deficit in humans, and many have heard about that. And here's an example.
Your name?
She's trying to say Laura.
Where do you live, Laura?
She's trying to say Sheffield.
And how old are you?
She's 4. So Laura's a 4-year-old child that has very decent, at least, speech comprehension, auditory learning, but difficulty producing learned sounds that she hears. And the difficulty is mostly in the sequencing of complex series of phonemes to make up words and imitate those sequences. And we, in collaboration with Simon Fisher, generated mice that have FOXP2, the same mutation that Laura has, and asked the question, what do these mice do? And we find that the wild type mice are able to switch when they see it, when they're in the presence of a female, they switch to producing these more complex sequences of vocalizations that the females like. Whereas the FOXP2 mutant mice just stay with these simple sequences here, shown at the bottom, a sonogram. And what about those layer 5 neurons? They exist in these mice. By the way, we don't think they have SLIT1, but they exist in these mice. And however, they're more spread out in the cortex compared to the wild type, indicating that the FOXP2 is actually controlling the localizations of these laryngeal motor cortex neurons into the primary motor cortex. And here is an example showing you that when you ask a female mice which speaker you like more, playing the songs of the sexier songs or versus these simple ones, the females spend more time next to the ones that the males switch to when they actually see her. And so to conclude here, what we've learned over the years, and this is a summary of the field overall, not just work in my lab, is that even though there's a lot of convergence here between humans and songbirds, there's a lot of similarities here that even though it's convergent, the mechanisms of that convergence are more similar than one would have imagined. They're not identical, but a lot of similarities, which means taking the songbird diagram here of the motor pathway for producing song and the learn motoring pathway for learning how to imitate those songs, down to the single neuron levels with the known timing from my physiologist colleagues about the timing of these firing rates and so forth for imitating those sounds, we can predict the cell types in the human motor cortex, Broca's area, other regions that correspond to these cell types here in songbirds. And this is where I think the field will be heading. From an evolutionary perspective, if humans and songbirds can evolve similar brain regions and circuits over the last 300 million years, and even amongst birds, amongst the last 65 million years, who's to say that a crocodile, a lizard, or another mammal, or even a non-human primate, in another 500,000 years won't evolve something the same thing. And when it does, I think I can say what that circuit in the genes would look like. So I will end off here saying that complex behaviors can evolve multiple times from deeply homologous but diverse brain circuits, cells and genes. And a complex trait like spoken language really, I think, can be understood from studies in distant related animals. The basic science of it, as well as the disorders of such traits. And that convergent evolution of mechanisms for a trait can be associated with similarities and disorders of that trait. I think these are high-level messages, I think, don't just pertain to the subject matter here, but all of biology that is being supported by NIH and others who are interested in disorders. And so I gave credit to the folks throughout, and I just want to show you their names here again, my team at Rockefeller, and also the collaborators I had and the funders that I've had, including from NIH, including shown in stars here are the funding support I had, including an NIH Director's Transformative One award we used on the Mouse Project and other things that I talked about today. And I'll stop and take questions. Sorry for going 5 minutes over time, but we're still under the hour, and I hope to answer your interesting questions. Thank you very much.
Thank you much, Dr. Jarvis, for such an interesting talk. We do have one question that I have see that's in the chat, and I want to remind others, if you have a question, please pop that into the Q&A box, and I'll read it off. The question is, efforts to create digital tools evaluating speech and language raise concerns given different dialects or word usage, potential to false negatives and positives. How can we avoid potential missteps given that they are slated for decision-making within healthcare?
So I guess this would be missteps in trying to genetically manipulate some animals to have some of these vocal communication behaviors that we have. I think that I'm hoping I'm interpreting that question correctly, and if not, then let me know. I think it's taken millions of years to get where we're at now, and evolution has its natural missteps, like in FOXP2 mutations and so forth. And what I think those missteps that could possibly happen in these non-human animal studies could be missteps that you will find in the human population when certain genes go awry, like FOXP2. So, I think I interpret that question, and that's my perspective on that. Does someone want to say something?
Yeah, I have another question following that, and we'll see if that person is happy with that answer. But
Okay.
You got their question. But there's another question asking, are you planning to apply spatial transcriptomics or potentially CIT-seq transcriptomics and proteomic experiments as a full genomic complement to your foundational work with the in situ hybridization and actively singing zebra French bird? Much more specific question.
Yes, we're planning to do both kinds of things that she asked. That is spatial transcriptomics. We're working on that now, not just for songbirds, but we want to do it on human speech-brain regions as well. With the single cell work, you can identify the cell type, but you don't know exactly where in the brain it's located, who's next to who, and so forth. And so with spatial transcriptomics, right now you can measure 1 to 200 genes at a time, as opposed to 1 or 2 with double label and CT hybridization. You can't measure the entire transcriptome of thousands of genes, but you saw that that's basically several hundred genes that are specialized in these speech circuits. So that's enough to look at with spatial transcriptomics. And we're going to do that in songbirds, humans, and the other vocal learners. And we're going to do it in the basal state as well as in the singing state. And I'm ready for the next question.
And I think we've hit the 3 o'clock hour, so I'm going to go ahead and let Bill Riley give us a close. But I really enjoyed your talk, and I think it's very important work, especially showing how we need and how much you can get from using multiple model species to attack a problem and answer a fundamental question, especially when you're breaking down all the scientific dogma.
Well, thank you very much. And I'm sorry that I took a little over time. I threw in a few extra slides there that I wanted the audience to know about, but hopefully you got the take-home messages. And anybody has questions in the chat, I can answer you separately.
Great.
Or post it somewhere, Bill, that you think would be the place to do.
Yeah, Eric, that sounds great. If we have other questions from people who are on, if you want to send those to us either via chat or email somebody at OBSSR, one of us, we can forward them to Eric and get your questions answered. Eric, I want to thank you again. As Dana said, certainly addressing some of the existing dogma out in the field and sort of challenging some of that, which has been great to see. And also sort of being a human embodiment of the integration of neuroscience and genomics. So we really appreciate that work. So once again, thank you for your presentation today. This is the last OBSSR Director Series for 2021. We'll be picking up again in 2022, so we'll let you know about that as it moves forward. Once again, Eric, thank you for your presentation, and thank all of you who attended today. Much appreciated.
Thank you very much.
Uh-huh.
Bye.